This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. Previously we saw that when producing genetically engineered plants, there is a risk of horizontal transfer of genes between plants and other organisms. Such transfer is problematic, especially if a resistance gene is inserted in a bacterium, because it will result in the production of an antibiotic-resistant bacterial strain. To avoid this, a lot of effort is made to remove the reporter genes used for negative screening, or more benign reporter genes are used, such as the gas reporting system. But it is not just reporter genes that can leak into other species. Hybridization between domesticated crops and their wild relatives can occur in the field because of the possibility for cross-pollination. This is an issue for genetically engineered as well as conventionally bred crops. Such hybridization is particularly risky when crops and their wild relatives coexist in the same agroecosystem, as is the case with sugar beet, sunflower, wheat, or sorghum. But genes of domesticated crops can also be leaked on purpose. In 2001, tens of thousands of hectares with genetically engineered Bt cotton were discovered in the Indian state Maharashtra, even though the Bt cotton was not commercially available at the time in the country. An investigation showed that Bt cotton seeds, which are produced and licensed by Monsanto in the US, have been illegally imported in India, then used to produce hybrid Bt cotton by crossing them with local varieties, which were then sold to local farmers. The unauthorized spread of GM crops is problematic because without testing it in local environment, we cannot know the risk their introduction entails for local ecosystems. However, the biggest issue raised by this unauthorized introduction was the violation of the copyright and ownership rights of Monsanto, which created the original Bt cotton. The issue was addressed with a similar legal approach as the piracy of artistic content distributed by giant production houses. To solve the issue of gene leakage, the giants of genetic engineering proposed the introduction of Terminator seeds that, in addition to having improved traits of agronomic interest, have also been engineered to produce sterile seeds, which should prevent their unwanted spread into nature. This controversial commercial move was quickly withdrawn because the Terminator plants can still spread their pollen to fertilize other crops, whereas the farmers have to purchase their seeds every year to sow in the fields anew. So what can we do to avoid transfer of genes from domesticated to wild plants? Probably not much. Species hybridization, especially in plants, has been occurring on a regular basis, even without human intervention. We could, however, reduce the risk of creating super-invasive weedy species by avoiding to introduce resistance genes in the domesticated crops as well. On the long run, a more effective way to address the issue is to change farming systems so that more diverse and locally adapted crop species are grown. Thus, rather than trying to introduce commercial varieties that have been bred to fit the modern farming requirements of minimal effort for maximal production. Having locally adapted species improved for traits that do not represent risk if their genes leak in the environment certainly requires more farming effort, but guarantees a stable production over time regardless of the unpredicted variation in nature, and entails less risks of introducing unwanted genes in other living organisms.